What I can give you is the story of some of our wanderings through the question. Okay, how do we get a slide here? Okay, these are some questions relevant to the function of sleep, and they're questions that laymen ask us regularly. How much sleep do we need? How little can we get by with? Most efficient distribution, et cetera, et cetera. We really can't give good answers to any of those questions because they imply some knowledge of what sleep is supposed to do. If you don't know what sleep is supposed to do, you don't know when you've had enough of it or when you've had too much of it. And we temporize and we tell people to get enough sleep to keep from being sleepy, and that's good advice. It probably won't do anyone any harm. It'll keep people from getting in accidents, etc. But we really can't give good, solid answers to those arguments. And yet we do to those questions. And yet we do think that sleep has a function. Doesn't necessarily have a function. Some people don't think it has a function. Some people think it takes up spare time. But these are the reasons that we think sleep is important. Number one, it's ubiquitous among mammals and among reptiles. Insects, we don't know. We know that insects show behavioral sleep, but we don't know whether that behavioral sleep is simply a stereotypic form of rest or whether it's analogous or homologous to mammalian sleep. Whether celled sleep is so speculative that I'm sorry I put it up there. Rebounds after sleep deprivation. Sleep behaves homeostatically. If you get less of it one time, you get more of it another time. And finally, sleep is adaptively expensive. That is, when we're sleeping, we're not eating, drinking, protecting our young, making money, writing grants, publishing papers, etc. So we sacrifice an awful lot in order to sleep, and one would think that unless sleep were important for some reason, there'd be little reason for it to have persisted through evolution. How do we go about finding out something about the function of sleep? Well, we use the strategies that are the strategies of science. This is essentially all we can do in almost any field. Number one, we can describe the phenomenon, we can describe the features of sleep, but description alone is hardly a guide to what the target of sleep is unless we get corroboration with other evidence. We can stimulate the subject and see what makes the subject sleep more or sleep less. But stimulation itself provides ambiguous answers. As you can stimulate an animal, and you may be stimulating the mechanism by which the animal sleeps. I would hardly think that giving someone a barbiturate or any other hypnotic is increasing the need for sleep. It's somehow stimulating sleep mechanisms. Thirdly, we can see what the correlates of sleep are, who sleeps a lot, who sleeps little. And that strategy suffers from all of the ambiguities of correlation, what's cause and effect. Then we can deprive, and we'll taught an animal of sleep or a human of sleep, see what goes wrong. That has its problems also. If you deprive an organism of sleep, what you may see as a result is not so much the deficit that results from sleep as the compensations for the loss of sleep, so that many of the performance deficits we see after sleep deprivation in humans can be very parsimoniously explained as the subject is simply trying to get to sleep as a homeostatic mechanism. And finally, when all of the above don't work, we lie, also known as theorizing. And the best theory should parsimoniously deal with these several kinds of strategies. Each strategy has its limitations, as we've seen, but fortunately the limitations of one strategy are not necessarily visited upon the other. The trick is to parsimoniously organize the data from several strategies to see if you can come up with a satisfying answer. Okay, now going to the descriptive features of sleep. Here are some of the descriptive features of non-REM sleep, which you're largely familiar with. And by and large, they suggest a toning down of activity, both systemic activity and cerebral activity. And there are signs of some activation as increased release of some hormones. You're all familiar with the increased release of prolactin and growth hormone. And in some brain areas, namely basal forebrain, you get an increase of the, quote, sleep-active neurons, which probably reflects the mechanism. But by and large, the features of non-REM sleep speak for a slowdown of the brain and the body, and it's a basis for the widely held belief that sleep is somehow for rest. And while there's a certain uniformity to this result pointing in the direction of slowdown, in terms of magnitude, the effect is not so great. That is, if you look at all the energy savings from the systemic reduction, reductions in systemic activity, it's been estimated that you only have about 5 to 10% reduction in metabolic rate during sleep compared to quiet wakefulness. And why would one suffer the indignities of losing vigilance just to get a 5 or 10% reduction in metabolic rate, which some people have estimated could be compensated for by eating a hot dog bun? And if you can do that, why waste a night sleeping? In terms of cerebral activity, most units in the brain slow down when you go to sleep, but they don't really slow down compared to quiet wakefulness. They slow down compared to active wakefulness. But as was shown by Evarts and Huttenlocher and Ubel back about 30 years ago, there's no overall slowdown in brain neuronal firing in the pyramidal tract, in the occipital cortex, in the reticular system during sleep as compared to quiet wakefulness. So while the descriptive features of non-REM sleep point in one direction, the magnitude of the effects are not very great. We go on to the descriptive features of REM sleep, and this is a real potpourri. I mean, if somebody can come up with some parsimonious theory of sleep, which includes penile erections, decrease in temperature regulations, increases in neural firing in 50 parts of the brain, decreases in 5 parts of the brain, et cetera, et cetera, et cetera. If you can come up with a parsimonious explanation of that, you're a better man than I am, Gunga Din. As a matter of fact, it seems almost as if every descriptive feature of sleep of REM sleep has inspired some theory of REM sleep. For example, REM sleep has rapid eye movements, so we have the theory that REM sleep is for oculomotor tuning. We have dreaming during REM sleep. It promotes the theory that REM sleep is for emotional expression or problem solving. We have increased brain temperature, so REM sleep is for brain warming. We have increased neuronal firing, it's for brain activation. We have decreased locus ceruleus firing, so REM sleep is for the upregulation of noradrenergic receptors. Namely, for every feature of REM sleep, somebody's invented a theory. This is not to say those theories are wrong. It's just to say that these descriptive features don't lead to any parsimonious theory. Okay, let's move to the realm of stimulation. This is just by no means a complete list, but this shows something of what increases or decreases REM sleep or non-REM sleep. And it's a real potpourri. If you can say anything about, if you can draw any parsimonious conclusion from those lists of stimuli, again, you're a pretty good generalist. If we go to the the major variables, the ways in which wakefulness, for example, may be intensified or de-intensified, we find quite surprisingly little effect on sleep, so that there's no effect of exercise on total sleep in animal and in human studies. There's no effect of stimulation. There's stimulus bombardment or stimulus reduction on total sleep. And in one of my favorite experiments that was done by Fredrickson and Hobson, a real classic, stimulation of the reticular formation electrically for a period of 3 hours resulted in no subsequent increase in sleep. In other words, there was no evidence that fatiguing the brain by overacting it increased sleep. There was a small increase in REM sleep, probably due to the fact that they were stimulating in the pontine reticular formation. Well, the fact that sleep is not very much affected by the conditions of wakefulness should give us some pause to consider the old, quote, truism that sleep is the price we pay for wakefulness. We don't know that sleep is the price we pay for wakefulness. We sort of accept that intuitively. But we may sleep simply because we need to sleep, not because we've been awake. And evidence that sort of suggests that direction are the recent results on ground squirrels, which, when awakened from hibernation, go to sleep. Certainly hibernation is not an awake state, and yet these animals apparently have a need to sleep that has accumulated during the hibernating state. So we don't even know that sleep is in some way a price we pay for wakefulness. Okay, now we go to the correlational data. By far, the best correlates of sleep are across species and with maturational variables. These data I'm going to report are abstractions from the results of Harold Zeppelin. I think that if anyone wants to study the function of sleep, if a student wants to get in that area, the place to start is to read Zeppelin's chapter on mammalian sleep in the Krieger Handbook. Zeppelin has had as many smart things to say about sleep as anybody who's ever touched the subject, and I urge you all to read it. This is a condensation of Zeppelin's data, and these correlations are shown in various degrees of highlighting. There are two major correlations that appear. And I say they're major because all the other correlations are either insignificant, that is the ones in regular type, or even the significant ones in small bold type drop out when you do partial correlations to correct for either body weight or brain weight. The two major correlations are between REM sleep, between body weight and non-REM sleep. Namely, it's a negative correlation, minus 0.62, smaller animals sleep more. The second major correlation is between brain weight and cycle length. Small animals have shorter cycles, non-REM cycles, than larger animals. The correlation between lifespan and cycle length is really determined by the correlation of brain weight and lifespan. So there are two major correlations here that are responsible for most of the variance in the other correlations. That's between body weight and non-REM sleep and cycle length and brain weight. The third major, this just illustrates it, giving the names of some real animals, showing the relationship between daily sleep time and body weight, with the little brown bat sleeping 20 hours a day and the big elephant sleeping 3 or 4 hours a day. Now, in considering the variables associated with REM sleep, there are three variables, all relating to maturational status or reproductive variables. This is the famous Rothwerk sign, showing that REM sleep reduces over the lifespan, with the greatest reduction coming in the early years. So younger animals have more REM sleep. And this is true not only within a species, but immaturity relates to REM sleep across species. This is a famous slide from Jouvet-Mounier that shows that animals born immature, and the rat and the cat, have a lot of REM sleep. Animals that are born relatively mature, like the guinea pig, have little REM sleep. And finally, there's a third set of variables that shows that immaturity at birth is related to adult REM sleep. That is, even when they're adults, animals that are born relatively immature have a lot of REM sleep. Well, what do we make of these correlations? They're very powerful correlations, and yet, because of the ambiguity of correlations, we don't know what function it is that these correlations reflect. For example, the correlation between sleep time and body size suggests an energy function, an energy savings function, because small animals need more energy. They require more energy to thermoregulate and to locomote, and also they can tolerate shortages of energy for a much shorter period of time than larger animals. Namely, we can go 16 days without eating. The little shrew has to eat about his body weight every day to stay alive. But what does this reflect about the target, or how might this come about? One would think, well, maybe this reflects a metabolic savings, and that's probably not the case, because, as I mentioned before, metabolic rate is reduced only about 5% during sleep. And secondly, in the Zeppelin correlations, if you correct for the contribution of body size, the relationship to metabolic rate becomes negative. Namely, it's the animals with the high metabolic rates that sleep less. They don't sleep more once you correct for body size. Another possibility is that that sleep may save energy, and save it more importantly in the smaller animals, simply by enforcing rest. Oh, we're going backwards here for some reason. This is taking off on its own. I guess they wanted me to hurry up. Okay, go to the REM sleep immaturity correlation. What's the functional significance of this? REM sleep may be related to immaturity because, as Rothberg and Demented proposed, it helps to mature the brain by activating it during REM sleep, namely activating it while the infant is motorically inhibited, so it doesn't fall out of the nest or the crib when the brain gets activated. It might be associated with immaturity because, as Jouvet has proposed, that innate neural patterns are rehearsed during REM sleep. It may be related to immaturity because it shifts the energy demand for heat to the parent. Namely, during REM sleep, thermoregulation is suspended and the parent provides the heat, and so this may save energy in the immature infant that would go into growth. Then, of course, there's also the possibility that REM sleep immaturity correlation is based on mechanism rather than cause. Namely, it may be the effect of an incompletely developed brain. That is, in the immature animal, it may have a developed brain stem which contains all the structures necessary for REM sleep, but it may not yet have a well-developed thalamus and cortex which would permit the emergence of non-REM sleep. So it may not reflect the need so much as a developmental condition. And finally, the third major correlation, the cycle length brain weight correlation. This is the most powerful correlation of all, a 0.88 correlation. It's amazing when you consider that these data were gathered by a number of different investigators, in a number of different labs, there must be a lot of error variance thrown in, and nevertheless you get that 0.88 correlation. Nobody's really ever pursued that. That's the rationale for that correlation. There are a couple of leads. As Zeppelin's pointed out, animals with small brains tend to have higher cholinergic activity and higher cholinergic acetylcholine content in the cortex, and so it may be a rapid turnover of cholinergic synthesis and metabolism that's responsible for the short cycle length in small-brained animals. Another possibility is that in the small-brained animals, there's a decrease in the density of neurons per unit volume of cortex. So in some way, a densely packed brain may have to have more frequent REM periods, perhaps to dispose of the wastes in this compacted brain. Nevertheless, these leads were suggested some 25 years ago. I don't know that anybody's pursued it. Nobody's really given a good look at the mechanisms for the cycle. Okay, finally we go to the strategy of sleep deprivation, and because of time and narcissism, I'm going to talk only about my own experiments. Now actually this is what I was asked to talk about. The first requirement of a deprivation experiment is really a combination of a stimulation and a correlation experiment. You apply a stimulus to stop sleep, and then you correlate the reduction in sleep with some effect. And so it's basically a correlative study, and to make any interpretation at all about the effects, you should have some reasonable equivalence of stimulation in an experimental and controlled condition. Otherwise, you don't know whether the effects are due to the sleep loss or whether they're due to the differences in stimulation. Well, to deal with that problem, we developed this apparatus, which many of you know about. We have two rats that live on a plastic disc. Oops, sorry. What I wanted was a pointer. There we go. Okay. Let's say this is the experimental rat, and he lives on one side of the disc. And the disc is situated over a shallow pool of water, only about 2 to 3 centimeters deep. And the control rat sits on the other side of the disc, and both are monitored continuously. Now, as soon as this rat starts to go to sleep, the disc is automatically rotated at a very slow rate, very easy to negotiate the rotation of that disc at that rate. And the rat has to walk in a direction opposite to disc rotation, otherwise the rotation will carry it into the water. So that's how the rat gets deprived of sleep. Now the important thing is that this rat is not always trying to go to sleep, no matter how sleep deprived. That is, sometimes it's eating, sometimes it's drinking, sometimes it's grooming or exploring the cage, and so this rat is awake. And when this rat is spontaneously awake, the disc is still and the control rat is asleep. And what typically happens is with this procedure, we can deprive an experimental rat of sleep of about 90% of his sleep, and this rat loses only about 30% of its normal sleep. These are the individuals who participated in these experiments over the years. They're just the people in the lab. We've had collaborators from outside the lab. It's impossible to convey the amount of work that they've put into these studies. While the procedure is automatic, it requires attendance about 14 to 16 hours a day, that you've got to sit with that apparatus to make sure everything's getting recorded well, that the parameters are checked, et cetera, et cetera. and you can only run 2 pairs of rats at a time. So it's a very slow grueling process, and the people listed here have performed these tasks very faithfully, very diligently over the years. And it's really their work that we're reporting on now. Okay, what are the results of these studies? We've now deprived rats either of total sleep or of REM sleep. You can set the apparatus so the disc turns only when they're going into REM sleep. We've done this experiment in about 200 pairs of rats. In every case, whether your rat's deprived of total sleep or of REM sleep, the experimental rat dies without fail. No rat has ever survived unremitting deprivation of total sleep or of REM sleep, without fail. During that time, only 3 or 4 control rats have died for mysterious reasons. I suppose that happens even in the colony cage to a certain extent. Okay, the next major effect is that the rats develop these very peculiar lesions on their tails, just like holes in the skin. First, they may start out as hyperkeratotic lesions, development of a horny surface, but then they develop into ulcerative lesions, which are just holes in the skin. They don't bleed, there's no pus, and they're localized. They happen only on the tail and on the plantar surfaces. They're not due to friction, because these lesions come up mostly on the dorsal surface of the tail rather than on the ventral surface. They're not due to water immersion. We've tested that. We've scoured the literature. We've consulted with dermatologists. We don't know of any other lesions like this in the literature. We found nothing that resembles it. They're very peculiar. They sometimes occur, this is a striking instance of it, bilaterally along the tail, with a spacing that corresponds to the spacing of the vertebral segments. I point out this slide because this is such a distinctive feature of sleep deprivation in the rat that if you saw this feature in any other condition, you would be obliged to describe it immediately. And we'll get back to that. Next major effect is, well, first let me introduce you to this graphic format. Different rats survive different amounts. That is, the pathological processes proceed more rapidly in some rats than in others. And to show the regularities in the data, we have them graded by percentage of survival time. And when you do that, you get very small variances. Namely, it sort of equates survival time across rats. Well, this is a graph of what happens in total sleep deprivation and in REM deprivation. And here first we have food intake. Over the course of deprivation, food intake approximately doubles in both conditions. Solid line's the experimental rat. It increases a little in the control rat, which is shown by the dotted line. And probably because control rats suffer some deprivation. At the same time that food increase is doubling, body weight is decreasing. Now, there's a number of conditions that can cause that. We've checked them out. Oh, wait, this is one more slide, which is actually tactically very, very important, personally important. These rats suffer a change in appearance. You know, what a healthy rat looks like. This is a rat sort of midway through deprivation, begins to look pretty scuzzy. The fur gets brownish. You see the lesions. They lose weight. The fur bunches up in clumps. Now, why this is important is that recently, some well-meaning sleep researcher notified a newsletter that our rats lose weight even though they eat it more. So I got calls from by no less than a dozen women's and fashions magazines inquiring about using sleep deprivation as a way of eating as much as you like while you lose weight. Well, the thing that stopped them from further inquiry is to point out that extended sleep deprivation will also make you ugly. Getting back to the changes in food and weight. Well, they don't result from dehydration. Done a number of procedures to check that out. It doesn't result from a malabsorption syndrome, namely the food isn't just going through the rat indigested. In fact, the food use is accelerated, and finally, by a variety of tests, we find that the metabolic rates are increased. They're increased by about 100% in the deprived rat. It happens both in totally deprived rats and in REM-deprived rats. REM-deprived rats die after an average of about 17 days of deprivation. The animals selectively deprived of REM sleep, or paradoxical sleep as we call it here, survive about twice as long. They die after about 5 or 6 weeks. The curves of the metabolic rate when equated for percentage of recording time are similar. Well, because we've traced this down to metabolic rate, for the remainder of the studies, we've used a measure of energy expenditure to describe these metabolic increases. This is a measure derived from the caloric value of the weight change and the food intake, and it's a pretty good way, a much less expensive way of estimating metabolic rate over time in a lot of animals. And as you can see, both in total sleep deprivation and REM deprivation, metabolic rate approximately doubles. Now, what's responsible for the increase in metabolic rate? Well, the first thing one thinks of is that there's some calorogenic hormone is run amok. And so we looked at several calorogenic hormones. We found that contrary to expectations, the thyroid hormones were reduced. Epinephrine was up, but by far the most important increase was in norepinephrine. And that happened in every rat, and it was a dramatic effect, which of course leads to the hypothesis that the problem may be that the sympathetic system is superactivated. And to test this hypothesis, we put the deprived and control rats on guanethidine, which is a peripheral sympathetic blocker. And with this, we successfully held down the increase in norepinephrine that results with the sleep deprivation. However, it didn't hold down the increase in metabolism, as energy expenditure went up as much as it had before. But what also went up was epinephrine. Instead of norepinephrine being showing the big increase, it was epinephrine that showed the big increase, which made it look like the problem was not a runaway calorogenic hormone. The problem was that there was an increased need for energy, and that the animal was going to use whatever hormone it could to generate the necessary metabolic activity. Well, if there's an increased need for energy, what produces it? Here's the more energy coming into the rat. The rat doubles its food intake. It's certainly not going into storage because the animal's losing weight. It's not going into waste, as bomb calorimetry showed normal or even accelerated food use. It's not going into work. With various strain gauge measures, we measured motor activities in these rats, and there was no increase in deprived rats compared to control rats. It must be going out as heat, which is a major vehicle for energy loss. And so we looked at temperature in these rats, and what you see in total sleep deprivation is an initial increase in temperature and then a decrease. In PS deprivation, there's only a decrease over time. For the moment, let's forget about this initial energy, the initial temperature increase, and consider only the decrease. This decrease is remarkable because this animal's dropping its temperature when its metabolic rate is doubled. And so it must be losing heat at a tremendous rate. And seemingly it would appear that the increase in metabolism is a way of compensating for the increased heat loss. Well, to test that idea, we made rats hypothyroid by putting them on a diet of propylthiouracil and made them more hypothyroid than they were ordinarily in the experiment, and then looked at what happened to their temperatures. That's by making them hypothyroid, we block the increase in metabolism. And here's what happens. This is it shows individual temperature graphs over the course of deprivation in euthyroid rats. And there's the usual initial increase in temperature and the decrease. If you block the energy increase by making the rats hypothyroid, temperature falls out of bed. It drops much sooner and goes much further, which is consistent with the interpretation that the increase in energy is somehow to compensate for the loss of heat. Now, what about the initial increase in energy? When we ran rats with hypothalamic temperatures as well as intraperitoneal temperatures, we found that the increase was even greater in the hypothalamus. And since theoretically, at least, and in fact it's widely accepted that brain temperature is held closer to the set point, better protected than the body temperature, it suggested that these animals had an increase in temperature set point. And we tested this as follows. We put animals in the thermal alleyway. There's just a long alleyway, which ranged in temperature from 0 to 60 degrees temperature. And this shows the animal's temperature choices over the course of deprivation. The darker bars are the control animals, and the lighter bars are the experimental animals. And what happens is that over the course of deprivation, the animals choose warmer and warmer spots in the alleyway. As a matter of fact, by the end of deprivation, the animals are picking 50 degrees centigrade, which is hot to the touch. No rat in its right mind would ordinarily go up to 50 degrees centigrade. Another way of checking on this is we provided animals with an operant. There's a hole in the cage, and if they stuck their nose in this hole, it would activate a heater which heated the cage. And when we did that, the animals increased their ambient temperature by about 10 degrees centigrade over the course of deprivation. So it looks like these animals have both an increase in set point and excessive heat loss, both of which create an increased demand for energy and causes the increase in metabolic rate. Because that initial increase in temperature doesn't occur in REM-deprived rats, made us think that the increase in set point was due mostly to the loss of non-REM sleep. And this is confirmed in a group of animals that were deprived mostly of high voltage non-REM sleep. And in those animals, temperature stays above baseline until the very end when they die. And so the combined results suggest that the heat loss is due primarily to a loss of REM sleep. The increase in set point is due primarily to loss of non-REM sleep. Is a new effect we found showing another thermoregulatory change. Don't have time to talk about it much. But normally, this is the drop-off in temperature as the animal goes from wakefulness to sleep, and this shows successive epochs of sleep. During recovery from sleep deprivation, or during the course of REM deprivation, what happens is that temperature doesn't drop when the animal goes to sleep. It's a rather dramatic effect. We've seen it in every rat we've run so far. As a matter of fact, I have to point out, I don't have to, but I'd like to point out that all the effects we've talked about so far, there's the increase in food intake, the decrease in weight, the change in increase in norepinephrine, the temperature changes, the norepinephrine change, the decrease in the thyroid hormones is true of every rat in which it's tested. These are not statistical, these are statistical averages, but they represent a change that occurs in every animal. They're very reliable effects. Well, this shows this sort of scheme of where we stand, namely the PS loss leading to heat loss, leading to an increase in energy expenditure, and leading to hypothermia. The animals become cold. But the hypothermia is not the cause of death. As if you keep animals in the cold and you drop their temperature even more, they don't die. The animals that are made hypothyroid didn't die any faster than the animals that had a slower decline in temperature. The cause of death is still unexplained. We don't know why these animals died. Recently, Everson has reported that in 5 of 6 rats that were made, that were carried to the point of impending death, these rats showed bacteremia, and this certainly reflects something important happening in these rats, but that's not a necessary cause of their death. If we block the bacteremia by giving the animals an antibiotic cocktail, these animals still die. The non-REM sleep loss leads to an increase in set point, also making for an increase in metabolic activity, and possibly creating a cachectic condition which might lead to death. However, this could only be a contributor of death, not the major cause, because Everson has shown that if you give rats a high caloric diet, they live about 30% longer than if you give them a normal diet. But nevertheless, they also die, even though they show little evidence of cachexis. So we don't know what's causing the death. And we also don't know what's causing the skin problems. So what do we know? We know that we've gotten, I forgot about this slide. This shows the effects that we don't get. Don't get the impression from the effects we've described that this is simply a product of the animals going to pieces, decaying in front of our lives. There's lots that doesn't change. There's no sign of liver or kidney malfunction. There's no indication of impaired immune function. There's no change we've been able to detect in the brain, either by looking at levels of amines or through electron microscopy studies or through studies of cholinergic and noradrenergic receptors. Something's got to be changing in the brain, but so far we haven't been able to detect it by any of these studies. We're just getting into some studies which will give us an idea about axonal degeneration using cupric silver stain, as suggested to us by Jerry Siegel, but we don't have any results on that yet. But so far, we haven't found changes in any of these parameters. So the changes I've talked about are rather specific effects of sleep deprivation. All right, now one of the obvious questions is, what's the role of stress in all of this? Well, the problem here, of course, is how you're going to define stress. You can define it in a conventional way by the activation of the HPA axis, looking at ACTH and corticosterone. ACTH is elevated just a bit in the deprived animals relative to control, but the difference is not significant. And the elevations here are really petty compared to what happens in most stress experiments, where you get 5 or 10-fold elevations in ACTH. If you look at corticosterone, there's no difference between deprived and control rats in PS deprivation. This difference is significant in the totally sleep-deprived rats. However, it's due as much to probably a chance drop in the control rats as it is by the increase in the experimental rats. If you compare the results, our results, that is what our animals show with Cellier's results, for example, there are a few spots in which there's a correspondence. That is, both animals, both groups of animals show an adrenal hypertrophy, but in the sleep-deprived animals, you see this only when they're near death, well after all the other symptoms have formed. In most other respects, the animals look very different. Cellier's animals don't die unless you keep them immersed in the cold for weeks on end or give them some very debilitating drugs. All of our animals die. The Cellier's animals showed increases or decreases in weight depending on whether they become hyperphagic or hypophagic. Our animals became hypophagic and showed a decrease in weight. It's quite different. And finally, Cellier described no skin lesions like the ones that we've seen, and these would be obvious to any investigator. Another way of looking at stress is to sort of make inference from the stimulus situation. And so we compared our results with those of the most severe stress study of rats we were able to locate. There's a study by Parade done in 1964 when he gave 5 seconds of a tone shock combination at 5-minute intervals for 22 hours a day for 23 days. Again, his animals showed an adrenal hypertrophy like ours. None of his rats died compared to all of our rats. His rats showed an 8.5% decrease in weight. Our animals but they decreased their food intake by 45%. Our animals showed a weight loss of 17% with a food intake increase of 75%. And finally, he reported none of the skin lesions like the ones that we've seen. So either by response measures or by a state of stress inferred from stimuli, our animals, the symptoms of our animals were not stress symptoms. I'm not saying our rats were happy. I mean, these are miserable, debilitated rats, and they're rats that proceed to the verge of death, and it's a very difficult experiment to do, and it's even harder on the rats than it is on us. But the point is that the changes we get, that we see in every single rat, are not conventional stress changes. You can't attribute these results to stress. Okay, where does this leave us overall? Well, what we've shown is that sleep deprivation, at least in the rat, is absolutely lethal, and that sleep is necessary for life. We've also shown that sleep deprivation, at least in the rat, we don't know about any other species, is important for thermoregulation. How and why is a mystery. I mean, with all the thermoregulatory mechanisms that we have available day to day, what sleep adds above and beyond that is a mystery. It must do something for long-term thermoregulation, some kind of meta-regulation that is necessary over and above the immediate thermoregulatory mechanisms that animals have available. And those are the two major things that we've shown. Well, these are partial answers. If you look at these symptoms, what does that have to do with the decrease in systemic activity during non-REM sleep? What does that have to do with the correlation between cycle length and brain weight? What does that have to do with the relationship between immaturity and REM time? I mean, it's just another fact, another bit of theory to add, adding to all these other theories we have. For example, here's a list of theories of REM sleep. I mean, what do we need, another theory? So we're sort of stuck in this dilemma of having a lot of pieces of evidence. And the major message that I have is that Well, we started out saying that there are many strategies for looking for the function of sleep. We've completely failed in any attempt to get at a parsimonious answer. Now we're sort of at a crossroads. Don't get scared, we've been at this crossroads for a long time, and we're still churning along. Now we can persist with multiple theories of sleep phenomena. These are the theories that start with one function of sleep is. And from a practical perspective, the partial theories may provide vehicles for getting grants, publishing papers, keeping jobs, etc. These are not crass motives. I mean, if you don't stay in business, nothing else will happen. Secondly, maybe you can get lucky if you come up with the right theory. In other words, throw in another theory of sleep, the worst thing that can happen with you is the indignity of a little intellectual clash in the literature, and nobody's ever died from that. And you might get lucky and hit a big one. More importantly, the multifunction approach might be substantively correct. Sleep might have evolved several times to serve several different functions. Or even if sleep had a single origin, it may have persisted because it came to serve several different functions. There are precedents. No less an authority than Darwin commented, quote, "What can be more curious than that the hand of a man formed for grasping, that of a mole for digging, the leg of the horse, the paddle of the porpoise, and the wing of the bat should all be constructed on the same pattern and should include the same bones in the same relative positions." It may be correct that there are a lot of different functions of sleep. Now, many of us, if not most of us, are fond of saying, however, we don't know the function of sleep. Apparently, these 20, 30 different theories that are floating around are not satisfying to us. We think there's something big missing. And so we say the function of sleep is unresolved. We don't say it's been resolved 20 times. We say it is not resolved. And to this way of thinking, the partial theories might be correct in a sense, and yet may trivialize the issue. It would be comparable to saying that the function of respiration was to whistle, sing, and play the tuba. Now, there may be a single major function of sleep which parsimoniously accounts for the diverse, descriptive, correlative, and experimental data on sleep. Just as cellular energy may be variously recruited to generate warmth, enable locomotion, or feed growth, sleep might provide basic cellular resources or mechanisms that preferentially serve maturation in the young, temperature regulation in small mammals, or higher mental processes in adult humans. I have no respectable idea of what such a cellular process might be. Most likely, it is not obvious or obviously connected to sleep. Otherwise, it would have already been identified by this very bright and very opportunistic group. I mean that seriously. The search for a parsimonious, reductionistic explanation of sleep may require leaps of speculation beyond the guidelines of rational inference. Thank you. We have time for one easy question.
Hello.
That's it. Thank you.
Oh well. It's not on. I can't ask the question.